Okay, so if you haven't, what do I want to say? If you haven't had enough, okay, transport at this point, we actually, or if you feel like you've had enough, we actually have one more we have to talk about. And it's probably one of the most important ones we deal with because it's quite unique in terms of the amount of stuff we can move across the cell. It's also unique in terms of kind of going against some of the principles we've talked about a little bit. Not necessarily against the principles, but kind of against with what we've been referring to up to this point. Up to this point, we've been talking about, and kind of one of the pieces I want to remind you of is again going back to the understanding of solutes versus solvent. When are we moving solutes? When are we moving the solvent? Specifically, when we're talking about osmosis. Now, everything we've done up to this point in terms of transport, has basically taken place on its own. Now sometimes it had a little help when we talked about facilitated diffusion, but we didn't expend energy, so we haven't discussed the role of ATP, and we've been moving with the gradient. So we've always gone from a high concentration to a low concentration. And so now we're going to kind of flip that, and we're going to go into, well, what if? Okay, we need to move the other direction, therefore we need to move against the gradient. And so we want to move from low to high. So that's a big part of our discussion. So as we talk about transport of materials into and out of the cells, but now specifically we're going to discuss the active transport of that. So one of the relationships we made right at the beginning of this principle is we were talking about the principle of entropy, which basically says, as kind of our natural law, that we proceed spontaneously or without energy expenditure. We basically proceed from a highly ordered to a disorder. And the example we used was kind of stacking toothpicks or stacking bricks, where the greater likelihood is we go from stacked to unstacked, but not the other way around. But what we want to do is we essentially want to go the other direction. To basically overcome that, we have to put energy into that system. So if we want something to happen in that we're going to put it back together, so to speak, then we've got to put energy into that process. So diffusion, notice it says here on the first bullet, diffusion results because of entropy. Substances move from regions of high concentration to low concentration because it goes from order to disorder. If we want to overcome that, so we want to go the other way, we want to take something that may be disorganized and now organize it, well, it takes energy to do that. We can kind of think of that as it's a lot easier for your house, rooms in your house to get messed up than they don't clean themselves, I guess is the way to refer to it. It's a lot easier to leave the clothes on the floor and scattered around than it is to kind of put them away and put them back into the dresser and put them back in places where you might be storing them. So that might be an analogy that works for you to understand this. Now, from the energy source standpoint, our biggest energy source that we're going to talk about in terms of active transport is ATP. And it's our energy currency is how we've kind of referred to it. And later on in this unit, we're really going to beat you up in terms of the role, in terms of how do we create that ATP and in what numbers we create them and for what sources we create them. So we'll get that into a big part of that later on. Now also one of our energy sources that gets probably the confusing part of this principle is that we're going to use pre-existing storage of energy in a concentration gradient. And I think the thing I want you to think about there is think of when we, now this year, in our area, of course, it's not a problem, with storage of water because there's plenty of it to be had this year. But you think about the water storage that takes place, so the water behind the dam. We used that example a little bit earlier in transport. But we store water behind the reservoir. Now, if that dam breaks, does it release a great deal of energy? Yes. So we have all this stored up water that basically has potential energy if we were to release that energy. Now sometimes we do. We release that out of the dam, we run turbines, we generate energy, we generate electricity. So we do that. But if the dam was to break, then there's a huge release of a large amount of energy. And so that will come into play with something we talk about here as we go along. Now, primary active transport. And I'm going to contrast that a little bit here in just a minute with what we call secondary active transport. So when we talk about active transport, as soon as you hear that term, couple things you immediately know, just like when we talked about passive transport. You immediately know that you're expending energy. You immediately know that your desire is to move a substance from an area of low concentration against its gradient to an area of high concentration. Now, to kind of tell you, and we'll beat you up with this for basically the rest of this class and the subsequent class you'll take, the Biomedical Core 2, because it's a very important physiological principle. the environment of our cells is such that we need to have the majority of the sodium on the outside of the cell. So it's our primary extracellular cation versus potassium inside the cell. It's our primary intracellular cation. And we need to maintain that environment. And so we're constantly moving sodium outside of the cell and potassium inside the cell. Now think about that. If most of the sodium is outside and we continue to move it outside, that means we're going from where sodium is fairly scarce, inside, to where it's fairly prevalent, outside. So we're moving from an area of low concentration to high concentration. The same thing is true with the potassium. We're moving potassium from outside, where there's relatively little, to inside, okay, where there's a great deal. So we're moving it to maintain this environment that we need, and we're going to use this environment to allow nerve cells to send information down the length of their cells. We use it to contract muscle. We use it basically in all of our cells. We maintain this environment of more sodium outside and more potassium inside. And one of the primary reasons for this is through the process of active transport. And we use what are called sodium and potassium pumps. And so you'll see this quite frequently. And so typically, again, our sodium is high outside, our potassium's low outside. Our sodium's low inside, but our potassium is high inside. And the reason this happens is our sodium-potassium pumps move those two ions against, you may want to underline that, against their concentration gradients, but they have to spend money, okay, they have to expend energy to be able to do that. They have to put in the effort. Now, one of the things I want you to pay attention to, and it's going to be a big part later on as we talk about metabolism, is pay attention in the book as you see these arrows going from one molecule, and then the products of that reaction generate the new molecules. That tells you the direction that equation proceeds. Now, in a previous module, we discussed basically the breakdown of ATP, okay, and then the buildup of ATP. Now, the reason that becomes important is anytime we put molecules together, anytime we form bonds, it takes energy to put those things together. It's kind of like, okay, if you're going to glue two things together, you got to put in the glue, you got to stick them together. And so as we form bonds, okay, we put things together. But now the nice thing is, if we put energy in to get those bonds to form, the good thing is we're going to get energy out. Because remember, one of the laws of thermodynamics says energy cannot be created nor destroyed nor lost. So the energy exists. We just change its form. So we can put it in as energy to form the bond, but when we break that bond, we get the energy out. So the energy's still there. It's just we've stored it, we've got potential energy, okay, and now we get it out. And so that sometimes becomes the confusing thing for students once in a while, is they say, well, ATP is energy. Well, yes and no. I mean, we can kind of refer to it that way, but it's better said that ATP is a molecule that contains energy, and if we break the bond, we can release that energy. So now I can transfer the energy from ATP to do something that I want, okay, which is to run this sodium-potassium pump, so that I can move a little bit of sodium outside and a little bit of potassium inside. So what happens here is, if we look at our normal gradient, and that's what the two arrows on the left are showing you, is my normal sodium gradient, if it was passive transport, would be that sodium wants to go this way, because there's more sodium outside than there is inside. My normal potassium gradient, if potassium was allowed to move, but it's not because it doesn't fit my criteria of small, neutrally charged, lipid-soluble. Therefore, because there's more potassium inside, its desire is to go this way. But that's not the way we want to go. We want to put a little more sodium outside and a little more potassium inside. So if we're going to do that, we have to use these sodium-potassium pumps. So we'll kind of go stepwise through this process. And so our pumps are going to be able to move these across. So step one in this process is that 3 sodium ions bind to the pocket, okay, in the enzyme, the transporter protein that we're going to use. And so 3 sodium are expelled from the cell, and ATP, okay, is split into ADP, adenosine diphosphate, plus a phosphate. But because we broke that bond, and that becomes the important part of this, because we broke that bond, now we release that energy, and that energy can then be transferred to our transporter to be able to move those ions across. That's what we're trying to facilitate. And so when we do that, it sets up the protein that then it exposes pockets for potassium. And so then we combine 2 potassiums, and then we can bring the 2 potassiums inside the cell. So what did we accomplish by this process? Well, with this process, we took 3 sodiums, moved those outside the cell against their gradient, and we moved 2 potassiums inside the cell. And so we facilitated that concentration difference between inside and outside. Now, one of the things we're going to get into later as we talk about the nervous system, one of the things we also did is we created an electrical gradient as well. If you think about that, well, how many pluses did we move outside? When we moved 3. How many pluses did we move inside when we moved 2? Well, okay, we're running a deficit. That's kind of like the analogy I use in class is, let's say you're playing slot machines in Wendover, Vegas, wherever you decide to go, and you decide to put 3 quarters in the slot machine. You pull the handle, 2 quarters drop out. Now little bells go off, or little lights go off, and so number one, you think you're a winner. Okay, now really think about it. You put 3 quarters in, you got 2 out. Okay, for the benefit of pulling that handle, you paid a quarter for that. So let's say you put in 3 more quarters, 2 drop out. Yay, you're a winner. Okay, no, you're not a winner. 3 more in, 2 more out, 3 more in. Well, eventually you're going to run out of quarters. So you're essentially losing. Now think about that in terms of our cell. If I've got 3 pluses moving out, but only 2 pluses moving back in, well, what kind of environment is that creating within my cell? Well, it's making the inside of my cell a little bit more negative, okay, at the membrane of that cell. And that's going to become very important to some of the things we talk about later on as we talk about, well, how do nerves talk to each other and how do we release neurotransmitters, and we're going to talk about some of that stuff. So that becomes important to us. So if you make sure you understand that. Now, I want to reiterate, I want to reinforce, what did we create? Okay, so I'm going to go back to the previous diagram. What did we create with what we did here? We created a situation where through primary active transport, through the use of ATP, we moved 3 sodiums out, so I've got a high concentration of sodium outside, and I moved 2 potassiums in, so I've got a high concentration of potassium inside. And we did that by moving those solutes against their gradient, but to do it, we had to release the energy from ATP, okay, to run our pump to be able to facilitate that. Now, what can we do to move other substances with what we did in primary active transport? So what we did with primary now gives us the opportunity to facilitate something else with the environment that we created. Now I'm going to go back to using kind of our water in the reservoir example. We expended the energy to create the environment where we dammed up all the water. And so we built the dam, we expended that energy. Okay, so that was our primary function, was to build that dam. And it took a lot of energy to do that. And the subsequent result of that is we dammed up all the water, and now we've created potential energy behind the dam. So we spent the money. Okay, we already made our initial investment, which created the water behind the dam. Now, with the benefit of the control valves, okay, we're going to utilize the energy contained in the dammed up water to facilitate other things that we want. And that's exactly what we do in secondary active transport. Now, in the process of secondary active transport, we use 2 unique types of transporters. We use what are called antiporters and also those that are called symporters. Basically, what we're doing here is we're moving 2 or more types of molecules. And the reason I mean 2 or more is in your kidneys you actually can move like 4 molecules at one time in some of these situations. And so 3 different types, but 4 total in your kidneys. And so we use these in our kidneys. We use these in our digestive system. And so this is not isolated in terms of where we use these. Now the names just tell us what these what these transporters are doing for us. So in this situation, our symporters are moving 2 or more ions, or 2 or more molecules the same direction. Our antiporters are moving them in opposite directions. You're like, well, how do we create this environment, or how do we do this? Now remember what we did, and the big player here is what we did with sodium. With sodium, we basically, kind of going back to our reservoir analogy, the plasma membrane becomes the dam, sodium becomes the water. In other words, did we dam up sodium ions? Yes, we moved all the sodium ions, well, not all the sodium ions, but we pumped the sodium ions with our sodium-potassium pumps. We pumped the sodium ions behind the plasma membrane, so to speak. Okay, we pumped it extracellularly, And because of the semi-permeable nature of the membrane, yeah, they can't go back inside the cell because they're not small, neutrally charged. So they can't move back across very easily. Therefore, we basically dammed them up behind the cell membrane. So have we created a high concentration of sodium outside? Yes, we have. Therefore, have we created potential energy that what if we can basically turn on the valve, have them come through, okay, like we would the dam if we want water to come through, run turbines, okay, create electrical energy, yes. And the same thing we can do with sodium. So what we're doing in this situation is we are using the established gradient of sodium that we established through primary active transport, and we're utilizing that energy that we created to run symporters and antiporters for secondary active transport. Okay? I hope that makes sense. So essentially the way this works is, for example, with the antiporter, we said an antiporter runs, okay, 2 or more molecules in opposite directions. And I really like the use that Dr. Hutchins put in here of using kind of our mill wheel here, that we're using our water wheel, okay, where we've created the environment of we've got sodium on one side, and we allow it to run down its concentration gradient. Now, I realize that doesn't require energy to do that, because we just open it up and sodium can run, but it took energy to do that. In other words, to dam up all the sodium, so to speak, on one side of the cell membrane, we took energy to do it, and that's why we refer to this as secondary active transport. It occurred because of, or it's able to occur because of primary. I hope that makes sense. So as we allow sodium to run down its concentration gradient, then it can basically lift calcium in this case, because these are actually called sodium-calcium antiporters. And so as we bring sodium down its gradient with our transporter, that protein brings calcium against its gradient. So it's an antiporter system. In contrast to that, sometimes we can move these molecules together. And so sodium, again, we drive it, that sodium comes along its gradient, and it basically, for lack of another term, it drags the other molecule with it. Glucose, for example, amino acids, in intestinal absorption of the amino acids that we do in our small intestine. This is exactly how, as you consume proteins and you break them down into amino acids, this is exactly how you bring them across from the lumen of your gastrointestinal tract, where your digesting food is, and bring them across into your intestinal cells. So you can absorb them in your bloodstream and then, of course, use them. And so it's a big part of what we do. So primary active transport, directly using ATP to run sodium-potassium pumps and to move sodium-potassium against their concentration gradient. Because of active transport, primary active transport, we can facilitate secondary active transport, which is moving ions still against their gradient. In the case, the examples we used were, for example, calcium and amino acids, because of the condition we created with primary active transport. Okay, so hopefully that makes sense. Okay, ask questions to your instructor as it applies.